Hey folks, this is Ethan Polliner with Cultra. In this video, we're going to explore the role that bacteria play in healthy soil systems. By the end of it, you'll know what they are, what function they perform, and why every productive field system relies on them. Let's dive into it. To understand why bacteria matter so much today, we must first understand where they came from and how they shaped the earliest soils. Bacteria were among the earliest life forms on Earth, emerging some 3.5 billion years ago. These tiny prokaryotes, with their simple cell structure and singular circular chromosome, were among the planet's first chemists. They learned to photosynthesize, fix nitrogen, and circulate carbon before plants and animals even existed. And that same biology still drives much of the ecosystem in our soils today. Bacteria are the most numerous and collectively one of the largest organisms in our soil. 500,000 bacteria can fit in the period at the end of a sentence. Yet they drive the majority of the biology in our soil systems. Almost everything a plant depends on can be traced back to something that a bacteria decomposed, transformed, or released for them. Now that we know how significant bacteria are, it's important to understand the function that they actually play inside of our soils. Bacteria come in three main shapes, those being sphere bacteria, rod bacteria, and spiral bacteria. But no matter the shape, they share the same strategy: reproduce quickly, turn over rapidly, and unlock nutrients within the soil. Under ideal conditions, a single bacterium could produce billions of descendants in half a day. Now in nature, bacteria rarely die of old age. Instead, they're often consumed by protozoa, nematodes, or are simply left dead due to shifting soil conditions. Now soil then is essentially a graveyard for bacterial remnants, and this turnover is what contributes to healthy and productive soil systems. Now moisture controls where bacteria can live. They must be surrounded by a thin film of water in order to move, expel waste, and absorb nutrients. And that means that when soil's dry, bacteria enter a state of dormancy, and when they re-wet, they reawaken just to continue the normal cycle. This dependence on moisture is one of the reasons that bacteria live closer to the root zone, where plant exudates contribute to a much more moisturous microenvironment. Inside every bacterial cell is a concentrated mix of sugars, proteins, ions, and metabolites. Membrane proteins pump nutrients inward and waste outward, contributing to a recycling of bacterial biomass. And these nutrients remain locked inside of that cell until it is eaten or dies. You can kind of think about these cells as little nutrient fertilizer bags. With a sense of how bacteria function, we can look at the key role they play in our soils: decomposition. Now, while fungi are the specialists in breaking down tough, woody materials, bacteria decompose fresher green residues that contain sugars and proteins. They release enzymes that break bonds in organic molecules, transforming long carbon chains into simple sugars, amino acids, and fatty acids. These compounds then become the raw ingredients for new bacterial biomass, maintaining nutrient cycling within the soil system. Bacterial nutrient immobilization is imperative to the health of our soil systems. Nutrients inside of bacteria cannot leach away. Only when they're consumed by a protozoa or a nematode do bacteria finally release the plant-available form of nitrogen, ammonium. This predator-prey mineralization loop is one of the primary pathways through which plants receive nutrients. It's also important to note that bacteria differ based off of their required oxygen levels. Aerobic bacteria thrive in well-drained, oxygenated soils and are the contributor for what gives soil its very earthy scent. On the other hand, anaerobic bacteria dominate when soils become oversaturated and oxygen deprived. These bacteria also provide us growers with an indication of what's happening in our microbiology. They're released scents with hydrogen sulfide, butyric acid, and ammonia. And these smells and compounds reveal when soil structure or drainage is failing. Once bacteria begin breaking down organic material, they enter the heart of their role in the soil: nutrient cycling. Bacteria sits at the center of carbon, sulfur, and nitrogen nutrient cycling. In the carbon cycle, bacteria break down dead plants and animals into CO2, which is then photosynthesized by the plant and converted back into biomass. In the sulfur cycle, aerobic bacteria oxidize sulfur into water-soluble sulfates, and anaerobic bacteria break down sulfur during decomposition. These transformations done by the bacteria shape sulfur availability for the plant. Now, the nitrogen cycle is where bacteria have their greatest impact on our soils. Nitrogen-fixating bacteria such as Rhizosphere and Rhizobium convert atmospheric nitrogen into plant-available ammonium. Other bacteria convert ammonium directly to nitrate through nitrification. And when soils become saturated, denitrifying bacteria turn nitrate back into atmospheric nitrogen, which becomes released from the soil. It's also an important note that bacterial activity depends heavily on pH neutrality. Most populations of bacteria will begin to sharply decline once pH drops below 7. To understand how bacteria survive and shape our soil system, we must take a look at one of their most important mechanisms: biofilms. Biofilm is a complex matrix of sugars, proteins, and DNA that bind bacteria to soil aggregates and bind those aggregates to the root tip zone. These biofilms help bacteria maintain their moisture, which prevents them from drying out. And it also allows for pathways for nutrients and waste. Within a biofilm, bacteria are significantly more tolerant to stress. This sticky matrix also builds soil structure. As biofilms cement soil together, it creates microaggregates for the bacteria to both bind onto and bind to the root tip zone. These microaggregates protect organic matter and create spaces for air and water to filter throughout the soil. Biofilms are therefore a survival strategy for bacteria and structurally valuable to the soil itself. Now that we understand biofilms, we can discuss where bacteria use them most aggressively: the rhizosphere. Plants release rich carbon exudates that bacterial biomass feed on. In exchange, bacteria mineralizes nitrogen and produces growth-promoting compounds. This exchange creates one of the most biologically active components of our soil system. Root growth itself releases cells and mucilage that bacteria quickly colonize. They remain close to the root zone, where moisture and nutrients are often dependable. When predators such as protozoa and nematodes consume bacteria in the rhizosphere, they release nitrogen exactly where plants are ready to take up nutrients. Plants also make use of a process called rhizophagy. During rhizophagy, root cells draw living bacteria inside of the root, they extract the nutrients, and they release that dead bacterial cell back into the soil system. When the root spits them back out, they regenerate and the process begins again. Rhizophagy tightens the nutrient loop between bacteria and plants and heightens microbial activity in the rhizosphere. Now let's understand anaerobic conditions and when bacteria might be pathogenic. When soil conditions change, the balance between healthy bacteria and anaerobic bacteria shifts alongside it. Anaerobic bacteria become dominant in compacted and saturated soils. These organisms produce alcohols, acids, and gases that are toxic to the plant roots. They also reduce the populations of bacteria that convert nitrogen into ammonium for the plant. This results in a heavy amount of nitrogen gas being lost directly to the atmosphere. Although these anaerobic bacteria still maintain ecological roles, they could be an indication that your soil is degrading in its structure and overall health. With all this in mind, we can now understand how biological inputs fit naturally with the already existing microbial populations in our soil. Nitrogen-fixing inputs convert atmospheric nitrogen into plant-available ammonium. Phosphorus-solubilizing bacteria release bound phosphorus. Residue digesters accelerate the decomposition of organic matter. And biocontrol strains produce antibiotics or trigger plant immune responses. Now, it's important to note that these products do not replace soil biology. Instead, they amplify and restore many of the naturally occurring biological mechanisms. Understanding colony-forming units, strains, formulation, and storage are all important to make sure that we are mimicking the naturally occurring microbial system within our soil. Understanding biological input compatibility is simply applying our ecological knowledge to crop management. And with all of these roles combined, we arrive at the full picture of why bacteria matter so much to the health of our soils. From the earliest days of life on Earth to modern agriculture, bacteria have shaped soil fertility as we know it. They decompose residues, cycle nutrients, and build soil structure, all promoting a healthy rhizosphere. Bacteria operate continuously and invisibly. For farmers and agronomists, understanding the role of bacteria is foundational to the health of our soil. The more we understand the biology beneath our very feet, the better equipped we are to build resilient soil systems that will feed future generations. My name is Ethan Polliner, the co-founder and COO of Cultra, a nutrient analytics platform for crop consultants and growers. Thanks for watching the video. We'll see you in the next one.